introduce your research in 45 minutes according to
45?
This is 30 minutes.
That's okay. That's
Okay.
Okay. Well, thank you for coming. I don't know how to do this. This one? I just want to think on how can I do the next? Yes. And there is one pointer. Okay. Great.
要不要拉窗？
And then I did 2 years in Kunming, China, or Xishanpana or Yunnan province. And I have done some independent, discrete researches. Today I combine all together and then make one story, which is titled Taxonomy, Systematic Evolution of Aquatic Wetland Plants in the Burma, Implication for Conservation. I hope you like this. And then this one is the table of content for the final report that I submitted last week. And I think some of you already checked. So today I summarized this final report and then follow this arrangement, like chapter 1 first, general introduction, and then chapter 2, chapter 3. And for some reason I explained some section 4.1 and section 4.2, and then complete with the conclusion, chapter 5. Okay. Oh, it's not good. Sorry, this one is chapter 1. So, for the general introduction, I should think about what I have been done, what my research, and how can I contribute to the conservation. The thing is, I belong to Center for Integrated Conservation, the XDBG, which is organized by Professor Richard Collett. So I always bear in mind how I can contribute to conservation. And then here's the thing. One of the important aspects at the center is this Zero Extinction Project, which aims to prevent extinctions in Xishanpana diverse native flora. And I'm pretty sure that this project is similar to the Worldwide Zero Extinction Project. And then what they say is to prevent species extinction by identifying the places where endangered species are restricted to blah, blah, blah. So today I explain what I have done here in terms of conservation. And then March 2017, this year, I joined a conference, which is ATBC, Association for Tropical Biology and Conservation, and Asia chapter meeting, Asia Pacific chapter meeting. And I found something, because in the keynote lecture, Tony said, science is the foundation of conservation. Then I realized that the conservation or conservation science is really huge project in which not only conservationists but also other disciplines like taxonomists should take part, like me. And then here I picked up some papers. Here is the taxonomy in conservation biodiversity. Here is the systematic conservation biology diversity. Here is diversity phylogeny conservation. So here's what I can do for the conservation and what I'm going to talk today. So, and then I have an area of focus because world is huge and I can't work for all the world species. So my focus here is the Indo-Burma biodiversity hotspot, which is one of the 32 world's biodiversity hotspot. And then, yeah, here it is. And I think Xishanpana is one of the part of this biodiversity hotspot, and probably Kunming as well. And then my species of interest is aquatic wetland plants, since I was a university student in Japan. And then aquatic wetland plants spontaneously appear on the phylogeny of angiosperm, as you can see here. But today I chose 4 genera, which belong to either Isomatares or Pores or Comerinares, all monopod species. Okay, now let's see chapter 2, species delimitation and cryptic diversity. And what you can see here is a new species that I'm going to describe, not yet describe, found in Vietnam, Vietnam. The aim of this chapter is to reveal cryptic diversity in widespread species. The species of interest are Monocaria vaginalis of Fonte and then Otteria erismoides of Hydrocaritis. And the method of choice are an orthodox molecular phylogenetic analysis as well as species delimitation in multi-species process. Okay, and then my species of interest here belong to genus Monocaria and then genus Otteria. It's not a big genus, like 10 species, and this one the genus of around 20 species in the world. And then the thing is, the both group is old world distribution, and the species are found in Asia, Australia, and somewhere in Africa. In this case, also some species are from Africa. But in these talks, for some reason, I focus on the Asia Pacific species. And here is distribution map of the the widespread species, Monocaria vaginalis and Otteria erismoides. The map is based on the Harvard specimen record, like in Missouri Botanic Garden or New York Botanic Garden, or Japan, Tokyo University or somewhere. So I think this dot mostly reflect that entire distribution of these species. And then my guess here is if this one is really one species or not, because these species are not only widespread species, but also species having extensive morphological, ecological, sometimes psychological variation. So let's see. And here is the workflow of this study. DNA sample synthesis, and plus the DNA and nuclear DNA markers. And then here is the orthodox phylogenetic analysis, maximum parsimony and then maximum likelihood and regime. And here is the Stacy species delimitation analysis. The reason why I did is to objectively assess the species boundary between the closely related species in the genus. And in the monographic work, we can find the key to species. So we use this key to species and then identify the specimen we collected. Okay. And then this slide shows the taxon sampling, the result of taxon sampling. I have visited many places like Japan, India, and Australia, and then collected 5 species of Monocaria and 7 species of Otteria. And of 32 specimens of Monocaria, 21 can be identified as Monocaria vaginalis, the widespread species. And in this case, of the 34 specimen samples, 20 samples can be identified as the widespread species. And the widespread species are shown here in the color red, like this one, from Japan to India to the Malay Peninsula. So let's see the widespread species. And then this one is the result of molecular phylogenesis of Monocaria, genus Monocaria. And then we obtained some grade from 1 to 7. And then what I want you to focus is this, the widespread species, which is not monophyletic in cluster DNA phylogeny and high frequency phylogeny, that divided into 3 grades. So this result clearly indicates that this species is not one species that have some cryptic diversity. So let's see. And this one is a species delimitation analysis. So what you see is to objectively assess the species boundary among species. So the best scenario is to discern species 1, 2, 3, 4, 5, 6, 7 species. So I named the 7 species, including 2 new species. These used to be the part of widespread species, carabin red. But now I split this widespread species into 3 species, including 2 new species. And here's the updated result. And again, we obtained some play, and then mostly the same between trusted DNA ITS trees. And then the thing here is, the important thing is the Ottelia erythmoides, the widespread species, is not monophyletic, and then divided into at least 2 plates. And then here, 2 samples place differently or inconsistently between the trusted DNA tree and ITS tree. I guess this is probably because the hybridization or grow plus capture. And here is the species delimitation result. And then in this case, the best scenario is to discern species 1, 2, 3, 4, 5, 6 species. And then the Monocaria erythmoides, the widespread species, is suggested to be 2 species, at least 2 species, and the Petiolata and the erythmoides sensitive. So again, I split the widespread species of the erythmoides sensurato into sensitive plus new species. So let's see what the species are. And this is one of the new species, Petiolata, from Vietnam. I collected in Ho Chi Minh. I mean near Ho Chi Minh. And then you can also see the specimen. This one is the holotype isotype. I think one of the types of the Monocaria vaginalis from India. And the point here is the elongated peduncle here, so that the flower overtop the leaf, but that is not like this. So this one is morphological characteristic as well. And the distribution is probably tropical Asian distribution. And at least we have 2 more samples from India and then Sri Lanka. And here's the second new species, Shudhastata. And believe or not, I collected this in Shishampana Tropical Botanic Garden. And I'm not sure if it's wild, but probably this one is a wild situation. And then I visited again last week and then found that the population is still good, a healthy population. It's good species. And the point here is this one is a multiflower, many flowered species, and somehow big in the body size compared to the Monocaria vaginalis. And we have, I have 3 more samples, and one is, I think, Malaysia, the other one from Singapore, and then the other one is from Thailand. So this one is, again, tropical Asian distribution, maybe a continental distribution. And here's the Ottelia, genus Ottelia, Ottelia petiolata from Thailand. I collected when I visited the 2012. And then, and here's a holotype of Ottelia erythmoides. I think described from India, again. And then the diagnostic character of this species is elongated petiole against the size of leaf, because this one is a big leaf and then elongated petiole, but this one is small leaf and then elongated petiole. And then we have only 1 sample from Thailand. But as far as I checked the specimen in the Bangkok Forest Museum, I think some specimens belong to this new species. And then the specimen are from Peninsula, Malay Peninsula. So I think this species, new species, is a Malay Peninsula element. But the thing is, these specimens are collected like 50 years ago, 100 years ago. It's really old one, and there's no new specimen. So what I want to say is, I have collected this one in 2012 in Thailand, but I have no idea if there is, if there is population exists in the Malay Peninsula or in the world. So let's summarize this chapter. I have found predictive overlooked diversity in Burma. And then the thing is that Monocaria vaginalis, widespread Monocaria vaginalis sensurato is divided into sensu-strict plus 2 cryptic new species. And in the case of Ottelia erythmoides, I split this species into sensu-strict plus 1 new species. And for the conservation status, I'm really worried about this species, Ottelia petiolata, because I as far as I know, there is only one locality in Thailand. So this one is in need of further survey. I mean, the field work. And then for the conservation biodiversity, what's the meaning of this study is just like this. Splitting taxonomy tend to increase protection, which is good. Because I split the sensilato species into cryptic species, novel species. So these cryptic species may have more attention for the conservationists or some people who care about the conservation. And then, yeah, so let's move on to the chapter 3. And what you can see here is the genus Nias. I think Nias kingi, from Thailand. And the chapter is for the systematics of aquatic and wetland plants. And the aim of chapter 3 is to revise systematics of widespread genera. And the general interest is Nias, of the genus Nias of Hydrocartici, and the genus Eliocarum of the Eliocaurici. The method of choice is not really specific. It's orthodox molecular phylogenetic analysis. Now, we explain some of the genus Nias first. And the genus Nias has some morphological variation, like the leaf texture and the seed coat, and leaf shoots, the female flower, and leaf surface, and male flower specimen. And the important character is the trait is like a seed coat morphology and the leaf shoots. Some species have a broadly rectangular shape, but other species have more like hexagonal shape. And here is the leaf shoots are rounded for these species, but some species have this kind of articulated morphology. So we use this kind of, and you also have some one-flowered species and two-flowered species, and the leaf surface has some, character, present or absent of some receptors or present or absent of specific.